Welcome to grade 12 biology. Today we are going to start grade 12 biology and we are going to start unit 1, which is about microorganisms. Microorganisms are tiny microscopic organisms that cannot be seen with naked eyes. They are very tiny, small, and most of them are unicellular. In fact, when we say most of them are unicellular, just you can understand that some are multicellular. Examples of groups, there are 5 main groups of microorganisms such as protozoa, fungi, some algae, viruses, and bacteria. Let's see one by one. Protozoa are unicellular microorganisms. They do not have cell wall. Most of them are motile. Most of them move from one place to another place. Most common examples of protozoans are amoeba, plasmodium, and paramecium. Students, remember that protozoans are examples of groups of microorganisms which are unicellular, which have no cell wall, and most of them are motile, even though some of them are sessile, cannot move from one place to another place. Amoeba is one example, plasmodium, and paramecium. The second group are fungi. Fungi, one of the most example of fungi is yeast, which is unicellular. All the others are multicellular. That's why we say most microorganisms are unicellular. Some species of fungi are useful. For example, Saccharomyces. This is used for baking purpose, in bakery factories for baking purpose. And some are in fact harmful. They can cause diseases. Candida is a causative agent of thrush in human beings. Many fungi produce mycelium. Mycelium is the reproductive structure of fungi. They release enzyme from hyphae to digest materials. Some are parasites. Others are saprophytes. What are saprophytes? Saprophytes are organisms that digest their food externally and absorb the products. This is hyphae, and this is my mycelium. This is fungi on orange fruit, and this is fungi on bread, and this, the bluish black that you see, is mold. So this is found on bread. The other is algae. The third group is algae. Many of them are seaweeds. Some are unicellular and the others are multicellular. Algae are used for food for fishes and other aquatic organisms. Algae produce more oxygen during photosynthesis than all of the forests in the globe. Some, in fact, unicellular forms like Chlamydomonas are moving from one place to another place, and they are motile. The fourth group are viruses. Viruses are intermediate between living and non-living because they cannot carry any processes common to living organisms independently. Mind you, viruses are intermediate between living and non-living. Why? Because they cannot carry processes common to living organisms, the characteristics of living organisms. For example, reproduction is one of the characteristics in living organisms. They only reproduce inside other cells. They are all parasites. They can even parasitize bacteria, and it's called bacteriophage. They don't have nucleus, they don't have also cytoplasm. They do have genetic material surrounded by protein coat. This is a virus, bacteriophage, a parasite bacteria, and the second one on the second picture is tobacco mosaic virus. This is a parasite on tobacco plant. They do have protein coats and nucleic acid and tail fiber. They do have the tobacco parasitizing virus have RNA core and protein molecule. Now let's proceed and see the last group of microorganisms, which are bacteria. Bacteria have prokaryotic cell. The difference between prokaryotic cell and eukaryotic cell is that prokaryotic cell have no true cell and have no true membrane or membrane-bound organelles. Their DNA forms a loop that's intermediate with the cytoplasm. They do have a cell made up of peptidoglycan, which gives rigidity, and cell membrane, cytoplasm, ribosome, and DNA, other organelles. But the cell wall that they have is made up of peptidoglycan. You know that plants have also cell wall, but the cell wall of plants is made up of cellulose, while the cell wall of bacteria is made up of peptidoglycan. This is an example of structure of bacteria, bacterial cell, structure of bacterial cell. It has flagella, it has cytoplasm, cell wall, capsule, chromosome, DNA, and cytoplasmic organelles. It has also DNA, plasma DNA, and it has a long tail. Bacteria are usually much smaller than eukaryotic cells because they do have prokaryotic cell. Their size ranges from 1 up to 10 micrometer in length. They are eukaryotes in between 10 to 100 micrometer long. They take photosynthesis, and this photosynthesis takes place in plasma membrane. This is one of the membrane in the cytoplasm, which is the powerhouse of living plants and other organisms. So photosynthesis in bacteria also, photosynthesizing bacteria, they do have plasma membrane. Many of the reaction, like respiration, takes place in the cytoplasm. Some have plasma membrane, and this respiration and photosynthesis is very important in bacteria. Now let's proceed classification of bacteria. Bacteria are classified in two ways. The first way is depending on the shape. Depending on shape, we do have three types of bacteria: cocci, bacilli, and spirochetes. Cocci or coccus have spherical bacteria, they have spherical shape. Bacillus or bacillus have rod shape, whereas spirochetes have spiral or corkscrew shaped bacteria. These are the three types of bacteria depending on shape of bacteria. So this is coccus, spherical, bacilli, rod shape, and spirochete. And these are the examples: bacterial cells and their shapes. You see the shape is spherical, and bacilli, rod shape, and spiral shape. The second classification of bacteria is based on whether it has gram-positive staining. Based on staining, we can classify positive and negative. Gram-positive bacteria stained and give purple by gram stain. It gives what? Purple color. Whereas gram-negative bacteria, when they are stained, they give pink color. So the difference is here the color that they give after staining. Gram-positive gives purple, whereas gram-negative gives pink color. So the difference is due to the structure of the cell wall of the different bacteria. So you can see gram-positive and gram-negative. This is the result of gram staining. This one is purple and this one is pink. Gram-positive, after the staining is finished, you get purple. Gram-negative, after the staining process, starting from fixation up to staining, the last process of staining, the result is pink color. So there are two ways of classifying bacteria. One is based on their shape, and the other is based on their color. So gram-negative bacteria have less peptidoglycan in their cell wall. We have seen that the cell wall of bacteria is made up of peptidoglycan, and in gram-negative, the number of peptidoglycan found on the cell wall is less than that of the gram-positive. And it is this peptidoglycan that absorbs the stain. Gram-negative bacteria also have a membrane outside the peptidoglycan cell wall, which is another cover, which gram-positive bacteria do not have. The other difference between gram-negative and gram-positive bacteria is that gram-negative bacteria have another envelope or covering outside the peptidoglycan cell wall. Outside their cell wall, they do have another membrane or covering. This outer membrane of gram-negative bacteria secretes endotoxin that helps bacteria to resist to antibiotics. You know that some bacteria cause diseases, and antibiotics are taken to overcome to the bacteria, And bacteria release what we call endotoxin to overcome this and resist these antibiotics. So diseases that are caused by gram-negative bacteria are more difficult to treat because they do have other covering outside the peptidoglycan cell wall. Gram-negative bacteria also cause more serious diseases, although there are exceptions. So this brings us to the end of today's lesson. Until we meet on the next lesson, goodbye.